Hey, how's it going, everyone? My name is Eric Singh, and I'm going to be talking about astrocytes. More specifically, I'll be talking about the normal functioning of astrocytes, as well as the process of reactive gliosis. And then Sebastian, he's our other educator, and he will be talking about uh, astrocytes and their contributions to neurodegenerative diseases and other CNS pathologies. But to get started, what are astrocytes, right? Well, for one, they are specialized glial cells that outnumber neurons 5 to 1 in the CNS. But what is their actual function? Well, that's actually kind of hard to explain because they do almost everything else that microglia and ependymal cells don't do. So, for example, microglia helps in the innate immune function, and ependymal cells help in regulation and the absorption of CSF flow in the ventricles and cisterns and whatnot. And the astrocytes really help do everything else to cell. And we'll talk about that later, including maintaining the environment, food, metabolism, everything like that. But going back to these slides, something that's really important that we have to talk about and really make sure that we understand is that each astrocyte, even though they outnumber neurons 5 to 1, and that there is a substantial amount of astrocytes in the CNS, each astrocyte has its own domain of control in the CNS. So even though there may be 10 astrocytes in one region, that one region is broken up into 10 separate areas, and each astrocyte, and there's one astrocyte per area. And that just makes sure that each astrocyte has control of the environment and it doesn't have to communicate with the other astrocytes to control that environment. However, there is some overlap, uh, mainly at the edges of each domain, and we'll talk about why it's important in later. And the reason why we need to know that that each astrocyte has its own domain is because in reactive gliosis, in severe reactive gliosis, actually, there's something called astrocytic proliferation, and this will present as many astrocytes in one domain and overlapping each other substantially. And I'll have a bunch of pictures and stuff to talk about that. And we've already talked about, the astrocyte is very important for supporting neurons in many ways. And there's many slides that will be discussing this in detail. And finally, a very important thing to talk about is that astrocytes, there is a gradated continuum of responses, which is mediated by specific signaling events. It happens in a spectrum and is not an all or nothing action like a neuron. Also, it's continuum, so it's not like this certain action creates this amount, and then you need a lot more to create the next amount. It's a little bit at a time, and it correlates with different CNS events. And this is all mediated by specific signaling events, which we'll talk about in detail. So what is the basic morphology of the astrocyte? Well, these are radially arranged, so they're coming outwards, these processes, and they come out in the shape of a star, which is why they're called astrocytes. And the reason why this is important is because this allows the astrocyte extensive coverage of the area that it covers. And as we'll talk about later, this is very important for maintaining the environment and other functions as well. And the astrocyte is able to communicate with all of the CNS, of all the tissues within the CNS. So this includes the neurons, the blood vessels, and because of this, is able to control the interstitial volume, and it also communicates with bordering astrocytes via the gap junctions. So I mentioned before, there is very little overlap in the domains, except at the very edges where there's communication between astrocytes via these gap junctions. There's four main types of astrocytes. One is protoplasmic, and this is the majority of astrocytes. And as we can see based on this picture, these are very close to blood vessels, right? So these are all different astrocytes, and they all have these little end feet processes going to this BV, this blood vessel. It's a very nice picture, a histological image to show what they do. And another thing that's very important is that this is found mainly in the gray matter between layers 2 and 6 in the cortical regions. And what this does is that it will envelop synapses so that it's able to control the synapses in the gray matter where the soma are. So other than enveloping synapses, it will actually communicate with these blood vessels via these end feet processes, and can affect the blood-brain barrier, as we'll talk about later. And because it has a very interesting function, because since it communicates between the synapses and the blood flow and the blood vessels, is able to synchronize blood flow with this neural activity. Now, the second type of astrocyte that we'll be talking about is fibrous astrocytes, and these are found mainly in white matter. And this has the least density of processes compared to other types. So what this means is I mentioned earlier that there are radially extending processes out of the astrocyte that makes it look like a star. However, as you guys can see in this picture, the green, the left on the left side, is a protoplasmic astrocyte, whereas the right side is a fibrous astrocyte. And you can see this has a lot more processes that extend radially, whereas this has a very few. Now, the reason why I really like this picture is because you can see that in the gray matter, there's a lot of somas, a lot of cell bodies, a lot of synapses, and you can see that this protoplasmic astrocyte is synapsing at these synapses. Whereas the fibrous astrocytes are found in the white matter. So this is where all of the axons are. And you can see that it doesn't necessarily bind to synapses, it mainly binds to the nodes of Ranvier. So it's very interesting, and it could also influence the environment and make sure that there's not too much of one neurotransmitter, as we'll talk about later. And as we'll see later, it also regulates the blood-brain barrier in terms of blood vessels and whatnot. So yeah. So the next two types of astrocytes, and I put them together in one slide just because they're very similar, just in different layers, as we'll see. So the first one is interlaminar, and it's called interlaminar because it moves between the lamina using vertical cable-like processes. And I'll also picture later on in the next slide to show this really well. But what this means is that the soma, or the cell body, originates in layer 1, but the vertical processes are in layers 3 and 4. Now, varicose astrocytes are basically the exact same thing, except that they reside in layers 5 through 6. And why is this important? Well, the current hypothesis is that the interlaminar and varicose astrocytes are essential for higher order cognition because they connect to different layers of the cortex. So when we think of the cortex, we have areas for vision and back, we have motor, the motor strip, somatosensation right behind that, and hearing in our temporal lobe, gustation in our insular cortex. However, to really integrate these functions and to have higher order recognition and better interpretation, we have to have connections between these layers and between these regions in the form of association areas. These interlaminar and these varicose astrocytes really help in bridging the gap and making these connections a lot stronger. So as you guys can see here, we have the interlaminar astrocyte right here, and the soma, as we said in the previous slide, is in layer 1, but these long entry processes go all the way to layer 4. And these varicose projection astrocytes are in layers 5 and 6, as we've discussed previously. And you guys can see the fibrous astrocyte by the white matter tracks, and then protoplasmic astrocytes, which are between layers 2 and 6. In this picture, we have some more histological staining of interlaminar cells. You can see how it has very large entry processes. These varicose projections are also very long radial processes. You can see another protoplasmic and fibrous. So I kind of touched on this a little bit previously, but how do astrocytes differ from neurons, right? Well, let's talk about how they're similar. So really, astrocytes and neurons, they both express potassium and sodium channels, and they can invoke inward currents. So they can bring in potassium, take it out, bring in sodium, take it out. However, neurons do this to create an action potential, right? So when you trigger the depolarization, sodium channels open because the extracellular is very high, intracellular is very low due to the sodium-potassium ATPase. So sodium will rush in, that positive ion influx will cause it to depolarize to positive around 30, 35. And then when it's at its highest peak, the sodium channels will close, potassium channels will open, and potassium channels have a very high intracellular concentration and a low extracellular concentration. So then they will leave very rapidly and cause the repolarization. Now, the difference is that astrocytes, they don't propagate action potentials, right? Neurons have an all or nothing response, where if they get enough excitatory postsynaptic responses, then they will create an action potential, which is an all or nothing, right? The only way a neuron can make a signal stronger is to have increased frequency of action potentials, whereas astrocytes have graded responses. So if they have a small amount of stimuli, then they're excited by a little bit. They have a stronger stimuli, then they're excited a little bit more, and so on and so forth. And astrocytes are primarily excited by increased intracellular calcium levels. And we'll talk about this later in terms of excitotoxicity and reactive gliosis and whatnot. So now we move into the next section. Well, same section, but now we're just going to talk, but instead of just going over the general aspects of astrocytes, we're going to go over more of the nitty-gritty of normal astrocyte function, talking about its role in development, its regulation of blood flow, its role in interstitial fluid homeostasis, influence on synaptic function, energy and metabolism, and then finally the blood-brain barrier. And I'll move on to the second portion of the presentation. So how do astrocytes help in development? So if you guys remember from the embryology video, I mentioned that we have radioglial cells that act as scaffolding and help the migrating developing axons to its location of where it needs to be. These glial cells, right, as we already talked about, astrocytes do everything that that the main glial cells don't, like microglia and ependymal cells. So these radioglial cells are really astrocytes, just a different type. So one, it helps it helps guide the migrating developing axons. Secondly, it also releases factors such as thrombospondin, which is essential for synapse development. This is actually also mentioned in my embryology video, wherein there were different petri dishes, one of just a few neurons and stem cells, another petri dish with neuron stem cells and astrocytes, and the one with astrocytes was able to create thousands of synapses, whereas the petri dish with just the stem cells was only able to create a lot fewer. So as we can tell from that experiment, it is very important in creating synapses early on via thrombospondin and some other release factors. Another thing is actually about in the embryology video is that it influences developmental pruning. So in the next slide, I have a nice picture about the complement system. So specifically, C1q is the initiator molecule for the classical complement system, and it's very complex. You don't really need to know the ins and outs of it. I just want to give you a nice picture to really conceptualize it. However, you will learn this more when you get to immunology and learning about the innate immune system. And finally, it is also essential to the development of white matter, so helping guiding these axons where they need to go, etc. So this is the complement system. It's very complex, but again, just you don't need to know the whole thing. Just kind of understand what's going on. So as we said, these astrocytes will release something called C1q. This is the initiator molecule for the classical pathway. And normally what this is meant to do is to target something called PAMPs, so pathogen-associated molecular marker. And what this will do is it will target these foreign objects and then tag it and then eventually kill it off. So as we go through this cascade, there's many processes that happen, and it's a cascade because they build off one another. And you have things such as cytokine release through which will cause inflammation, opsonization, which is tagging the foreign objects, which will make it easier to be bound and killed by other immune cells. More inflammation via cleavage of C5 protein, and finally cell lysis, which is the polymerization or the recruitment of C5b, 6b, 7b, 8b, and then finally polymerization of C9. I added this picture to show that you have C5b recruiting C6, recruiting C7, recruiting C8, which recruits C9, which will then polymerize into a nice, very large pore through the cell membrane. And this will cause cell death. One, because, well, the main reason is because this pore cannot be closed. So if, for example, you aren't able to control what goes in and out of your body, like in a stab wound, it's very likely you're going to die unless you close it. But if it's unclosable, like as in this case, then the cell will always die through cell lysis because of the environment and other things such as that. Now, regulation of blood flow. So these astrocytes, they release many molecular mediators, including vasodilators such as prostaglandins and nitric oxide, as well as vasoconstrictors such as arachidonic acid. Now, if we think about what those mean, right? Vasodilator, vasoconstrictor, right? Vaso means blood vessel. Dilator means to get bigger. Constrictor means to get smaller. We need to think about why is that important, right? Why not any? Why does the astrocyte affect the blood vessel diameter? Like, why is that important? And it's important because this can very quickly and very efficiently regulate the amount of blood flow to the brain. So, and I added this picture at the bottom to really explain why, right? So this equation is about fluid resistance, right? So resistance is equal to 8 times the length of the vessel times the viscosity of the fluid divided by pi times the radius to the fourth power. So a couple key, so you don't have to memorize this, but just understand what it means. It'll help you understand why we use vasoconstriction and vasodilators to regulate blood flow. So this means is that the length of the vessel, right, is the longer the vessel is, the more resistance you'll have, right? Which makes sense because you have more, it takes, there's a longer path for it to go. There's more resistance because of the friction along the walls. Viscosity of the fluid will obviously increase the resistance because more viscosity means more friction within the fluid flow. Now, pi, you guys know, 3.1415. And finally, the radius of the vessel, right? This is very important because this is to the fourth power. Okay, so to switch gears for a second, we need to talk about why or the signals the astrocyte receives to regulate blood flow. So as we talked about with the protoplasmic astrocytes, they have connections between the synapses as well as blood vessels. And this is very important because this allowed scientists to hypothesize and eventually prove that blood flow is closely modified based on neuronal activity because of these connections. So if we think about the neuronal activity, right, synapses are firing off very quickly, and a very quick action requires a very quick response to respond appropriately. So for one, if we want to develop a smart way, or if these astrocytes want to be smart and regulate blood flow in a proper way, one, it has to be acute to keep in pace with synapse changes. It also needs to be realistic. So I'll explain why. So it has to be realistic and acute. It has to be effective. So length of the vessel, the body can't really control that, especially in an acute sense. The only way you can change the length of a vessel, especially and even less so in the brain, is through angiogenesis. And this takes a significantly long time unless there is a cancerous form, cancer. So it takes a very long time, and of course, we can't grow vessels and break vessels down because of the continually changing synapse activity. We can't really change viscosity because viscosity of fluids, especially the blood and the plasma, is based on the function of the kidneys. So that if we want to change that, that would require the astrocytes releasing endocrine factors to the kidneys to either reabsorb water or secrete ions like potassium, sodium, and others, right, like ammonia and whatnot. But this will take a very long time to do, and this would not be effective if it's in response to synaptic activity. Now, the radius, the arc to the fourth. This is very important because the fourth power is very significant because this means that you can have a very tiny change in the radius. And if you take that to the fourth power, right, that will result in a very large difference in the radius, or in the resistance, sorry. So a small difference in radius in the radius will result in a very large difference in the resistance. So we don't need a lot of these molecular mediators to respond appropriately to the synaptic function. And if you guys know from your anatomy and physio that these arteries and these arteries, they have smooth muscle surrounding it. So it can very easily constrict or contract based on the molecular mediators that binds to its receptors, which will allow for a very quick and very efficient change in the radii of these vessels, and really suits the function of it. So the next function of astrocytes that we'll be talking about is interstitial fluid homeostasis. So one of the things that astrocytes have, especially the protoplasmic astrocytes, is that because of their very strong connection to these blood vessels via their end feet projections, they have access to a lot of water right from the blood and the plasma. So these astrocytes are very rich in aquaportin 4, AQP4, and if there's dysfunction, such as an excess, then it can cause vasogenic and cytotoxic edema, as seen in strokes and other pathologies of this fast-growing cancers. So as you guys can see from these CT scans, this darker region is all edema, right? This is all edema right here. Another thing that's very important is the uptake of potassium. So as we talked about in neurons, right, the action potential is very reliant upon potassium, especially potassium, more so than even sodium, because the membrane is very permeable to potassium due to the having a lot of these potassium leaky channels. So as we've talked about, in the action potential, the depolarization is sodium coming in to become positive 35 or so, and then you have a repolarization where sodium leaves. However, this is all based on gradients. So if you're trying to repolarize the neuron, then you would need the potassium channels to open. However, potassium will not leave unless the intracellular concentration of potassium is a lot higher than the extracellular levels of potassium, which allow it to move outwards towards this gradient. However, if there's excess potassium extracellularly in the interstitial fluid, then the cell won't be able to repolarize because the potassium won't move out of the cell. And this could be a huge issue because this essentially immobilizes or handicaps the neuron from making creating more action potentials. This can cause other issues, as Sebastian will talk about, in terms of excitotoxicity, because it thinks it's completely on when it shouldn't be. Another is proton shuttling, which is very important for pH regulation in the CNS, very important for protein folding and whatnot. So if your pH balance is off, it can affect a lot of intermolecular binding, affecting the structure of proteins, and then finally its function. This is seen especially in Parkinson's disease because depending on the pH of the environments, the alpha-synuclein can actually have a completely different structure and function, and potentially even accumulate into these fibrils, and which will eventually form plaques, just because it is a protein that has a very flexible structure, and differences in that structure cause very different functions, which in Parkinson's case is very detrimental. And another thing that it does, we talked about the protoplasmic astrocytes having processes at the synapses. So especially in the case of glutamates, what the astrocyte is important for is taking up the excess glutamates so that it doesn't overly excite the neuron, right? Because that can cause excitotoxicity, calcium toxicity, which leads to excitotoxicity, and lead to cell death. And then these astrocytes will then cycle through the glutamate, create glutamine. Glutamine shuttles back to the neuron, which can then convert that back to glutamates via a deaminase reaction, and then use that again later on. So very important there. And as we've talked about, networks of astrocytes via gap junctions rapidly dissipate accumulation of molecules to prevent potential accumulation. So again, this goes right back to the fact that there is very little domain overlap except on the edges where they're connected by gap junctions. So one of the reasons why this is important is because if you have one astrocyte having too much glutamate coming into its system, then it can increase intracellular calcium, which will then cause the death of the astrocyte over time. However, if you allow this calcium to diffuse through these gap junctions to different astrocytes, it can kind of mellow out a huge spike in calcium over a large area and prevent possible excitotoxicity. And the astrocyte has influences on synaptic function, right? So we've been kind of saying that astrocytes really help at the synapse, but we haven't actually said that astrocytes participate actively in the synapse. And recently, it was actually shown that astrocytes actually release synaptically active molecules, including glutamates, purines such as ATP, GABA, which is inhibitory in the spine, as well as D-serine. And as you guys can see from this bottom picture, right, the release is based on calcium oscillations because, as we said, the astrocyte is excited not by action potentials but by calcium oscillations. So there's a hypoactive state, a physiological state, and a hyperactive state. Now, there's something called the tripartite synapse, and this is a hypothesis which states that astrocytes play direct and interactive roles in the synaptic activity, which is essential for information processing by neuronal circuits. And this was really shown because in pathological states, right? And I'm sure Sebastian will talk about this more, but really quickly, if you have a hypoactive astrocyte, it can lead to symptoms such as schizophrenia, and when it's hyperactive, it can lead to even epilepsy. So it has very strong influences on the synapse. It also has long-term effects on synapses through the release of growth factors, which is used for synaptic remodeling of the adult brain. So as I mentioned in the previous videos, in the embryology video and in other PowerPoints in the developing minds, very early on, there's lots of growth factors that allows for the explosion of growth in the CNS. And in the adults, these growth factors decrease significantly, except in some areas associated with memory, such as the hippocampus, cerebellum, and some areas of the prefrontal cortex. And these growth factors are released by these astrocytes to really help. So energy and metabolism. So the astrocyte is really important for taking up glucose from the blood, right? As you said, the protoplasmic astrocytes takes glucose from the blood via these GLUT receptors, GLUT transporters, and will store it as glycogen. Now, these glycogen stores are used in hypoglycemic states where there isn't enough glucose in the blood, such as when someone is fasting, or when there's high neuronal activity and the normal glucose in the blood isn't enough to supplement the neurons during these levels of high activity. Now, something that's very interesting is that the astrocyte does not give neurons this glucose. It gives it a metabolite of it, which is lactate. If you guys know, lactate is similar to lactic acid when you're exercising during anaerobic metabolism due to an anoxic environment because your heart isn't able to get oxygen to those muscles. It's interesting how the astrocyte is in this picture. You can see that glucose will go by GLUT1 receptors, become pyruvate, go through glycolysis, and then become lactate, go through these lactate shuttles towards these neurons, right? Go through citric acid cycle and oxidative phosphorylation and become ATP for the neuron to use. So just interesting that the astrocyte does not give glucose, it gives lactate. And now a very fun topic because there's a lot of debate on what the blood-brain barrier is. But what is the blood-brain barrier based on the literature we have right now? Well, this impedes molecular influx into the brain parenchyma based on its polarity and size. So the major player of the blood-brain barrier are these tight junctions at these endothelial cells. And because nothing can really go through these tight junctions except for some water-soluble agents, really, these other proteins and molecules has to go through certain receptors and certain transporters to get through. Now, there is some evidence, and now this isn't just it, right? The endothelial cells aren't the only components of the blood-brain barrier. The astrocyte also influences the blood-brain barrier. It was shown in certain experiments where there's leakages that occur when the astrocytic end feet have dysfunction and be due to specific signaling mechanisms. However, there's some evidence against astrocytes regulating the blood-brain barrier because in utero, there's evidence of there being regulation of the blood-brain barrier before astrocytes have formed yet. So what new evidence has shown is that astrocytes don't necessarily they aren't the blood-brain barrier, but they influence it significantly. So the blood-brain barrier can function somewhat well without an astrocyte, but the astrocyte releasing certain molecules and signaling molecules is able to influence it and gain more glucose if needed, or different molecules like water through the endothelial tight junctions. So now we talked about a few of the normal functions of the astrocyte. It's important to talk about what happens when the astrocyte responds to CNS damages. Now, reactive gliosis is very interesting because there's no widely accepted category of what reactive gliosis is when it starts, the categories of it. However, I've come up with 4 factors that I found ubiquitously within the literature that can really help you guys really get a glimpse of what it is, and we'll build off of this and talk more about it. So there's 4 factors that I want to talk about. So 1, the term reactive gliosis is a spectrum of molecular, cellular, and functional changes occurring as a response to CNS injury. 2, the changes are gradated to the extent of CNS damage that is being responded to in a continual fashion. 3, these changes are regulated by intra and intramolecular signaling molecules. And 4, reactive gliosis has a potential to alter astrocyte function either through loss or gain of detrimental or beneficial function. So let's slow down a little bit here and let's talk about what this means. So when I say that reactive gliosis is a spectrum of molecular, cellular, and functional changes as a response to CNS injury, this means that this process called reactive gliosis can radically alter the astrocyte on a molecular level, at a cellular level, and this will affect the function of the entire astrocyte. And this is all in response to what happens to CNS. So because it is a spectrum of change and it is continuous, as you said before, then a very minor CNS injury will cause a minor change in the astrocytic function. The second, and we just talked about it, is that changes are gradated to the extent of CNS damage that's responded to. So that's exactly what I just talked about, where the changes are at an incremental process, where small CNS damage caused a small reaction of reactive gliosis. A lot of CNS damage caused a very severe effect, or it's very severe reactive gliosis. And 3, reactive gliosis is all based on inter and intramolecular signaling molecules. We'll talk about this in depth later on, but this is very important because the astrocyte is able to respond to specific CNS damage because these specific damages will release specific molecules. And then when the molecules are bind to the astrocyte, then they know that that certain injury happens and will be able to respond to it. Because a astrocyte can't tell the difference between a penetrating injury or a blunt injury to the head. And you need specific molecules, signaling molecules, to tell all the cells in the area what's going on. And I really wanted to talk about this, and I'll talk about this later on, is that in literature in the past, there is a lot of papers that have just blankly stated that reactive gliosis is detrimental and it is a bad function. However, there is truth to that, wherein it can cause a lot of damage. However, it also has very beneficial effects to the damaged CNS, as we'll talk about later as well. So the kind of the first stage, a very mild version of reactive gliosis. This is the kind of reactive gliosis in response to very minor CNS injuries. So some of the things that occur is hypertrophy of the soma, so the cell body of the astrocyte will get larger, and as well as the processes within their domain. So this is important because, as we'll see in severe stages, there is infiltration and severe overlap of astrocytic domains. And we'll talk about the issues with that later on. There's also an upregulation of GFAP. Now, the issue with GFAP is that historically, we have almost exclusively used GFAP to visualize these astrocytes. One, because reactive astrocytes typically can show GFAP, whereas normal astrocytes won't. However, a lot of other glial cells will express GFAP. So in past literature, in the very early papers, people have said that, well, in mild to moderate reactive gliosis, there was astrocytic proliferation, but that's only because GFAP was expressed in other glial cells. So it seemed like there was an increase in astrocytes when there really wasn't. Which leads to the third point, which is there's little to no astrocyte proliferation. And I'll show you guys pictures in a minute, which can really cement this idea. And as I said before, mild to moderate reactive gliosis occurs in relatively minor CNS damage. So mild non-penetrating or non-concussive injuries, innate immune activation, such as microglial activation, detecting foreign objects and immediately phagocytosing it and releasing some inflammatory cytokines, and also areas distant to focal lesions. So this is an important thing to discuss because even in things such as, let's say, a gunshot to the brain, the area immediately around the impacts will have very severe gliosis and scarring. However, in the other side, a few inches away, it will just be mild reactive gliosis because it's so far away, which leads to something important we'll talk about later, is that these changes via reactive gliosis is both temporally and spatially determined. And I'll talk about that more in depth later on. And finally, if the triggering damage is rectified or reversed, then the astrocyte can revert towards normal appearance and function because there's very little hypertrophy and there's no proliferation of astrocytes, which resulted in no major overlap within the domains. So the astrocyte can reverse reactive gliosis and go back to its normal functional state. So now to move on to severe diffuse reactive gliosis. So compared to mild to moderate, we have even more upregulation of GFAP. We have even more hypertrophy of the cell body. And in this case, there is astrocytic proliferation, which will also, and because there's this proliferation, this division of astrocytes, then these astrocytes will infiltrate other domains and cause significant overlap, as I'll show you in a second. And finally, because of this proliferation and this overlap in the astrocytic domains, there is long-lasting reorganization of the tissue architecture, which can have potentially long-term changes within the CNS. So to switch gears, look at these smaller pictures on the bottom, we can see that this is astrocytes that are remote from the lesion, right? So if you guys remember from the previous slide, there is the reactive gliosis is regulated by based on distance from the lesion. So very far from the lesion, we can see that there is one astrocyte right here, right, with some expression GFAP, but not very intense. And you see that around it, right, which processes are, there is not much overlap except for a little bit right here where the gap junctions are to help equilibrate the amounts of ions and molecules, right? So this is a pretty normal histological picture of an astrocyte. Now, in moderate reactive astrogliosis, we can see that the soma is a lot darker, and the processes are a lot thicker. You can see that they're a little bit closer, but there's still not as much overlap as we'll see in this one. You can see there's still some division, there's still some separation, right, from this one to this one, or this one and that one. There's still some division. We can still see some pretty sparse areas with no astrocytic processes. And finally, in this case of severe diffuse reactive gliosis, we can see that there's very massive hypertrophy of these cell bodies that are very close together because of this proliferation, and there's significant overlap in the processes of these astrocytic domains, which can cause this long-lasting reorganization of tissue architecture. So I really hope that these pictures really cemented the idea of the changes in the morphological changes of these astrocytes. There's another stage wherein there's severe reactive gliosis, as in the previous, but there's a added component of compact glial scar formation. So again, there's further upregulation in GFAP, soma hypertrophy, and there's substantial astrocyte proliferation, so that now there is no visible astrocytic domain. So even in the severe reactive gliosis, we can still see some areas that were a little bit differentiatable, but in this stage, there is very little. There is complete obliteration of these astrocyte domains, and this glial scar formation. It is a dense, narrow, and compact glial scars. And within this region, there is proliferation of other cells, not just astrocytes, such as fibromeningeal cells. And these are important in help in the deposition of a dense collagen in the extracellular matrix. And finally, they also forms along the borders of severe CNS damage, necrosis, infection, or autoimmune triggered inflammatory infiltration. So this says, going back to the previous example of a gunshot wound to the head, very far away from the impact would be mild gliosis, but along the border where the bullet entry path, you're going to have compact glial scar formation, where there's these astrocytes have completely different morphology, and they're very, and you see here, they're very tightly packed together. Then you have these fibromeningeal cells, and then the extra dense collagen within the ECM, which is a border to prevent spread of the traumatic tissue, possible ischemia, the cytotoxic materials, infectious agents, autoimmune antibodies, inflammatory cytokines, or even neoplastic materials so it doesn't spread. And you can see within this histological picture, we can see this is a very dense extracellular matrix. Processes everywhere, it's very dense. There's almost no differentiation between the somas, whereas in here, it's pretty bad. They're very close together, but there's a very clear delineation between the two right here. So you can very clearly see that. Now we've talked about all the stages of reactive gliosis. Let's talk about how it's triggered. So you don't only need to memorize this slide, just really understand what's going on. So these are inflammatory, so just really understand that many different molecules can trigger reactive gliosis. And as I said before, because certain CNS damages releases certain molecules, this really tells the astrocyte what kind of damage has occurred and what is the best response to it. Because as we said, because the morphological and functional changes to the astrocyte can be good or bad, depending on the time, the distance away from the injury, etc. So these are mainly inflammatory cytokines, which is from an infection, right? This here as well, the innate immunity of toll-like receptors and lipopolysaccharides from bacterias, many bacterias. I believe gram negatives, from here. And then excess neurotransmitters such as glutamate and hormones of noradrenaline. So we said glutamate, excess glutamate can cause excitotoxicity due to increased calcium concentrations. Purines such as ATP. So when there is injury to the CNS, death of these neurons will release ATP. ROS, reactive oxygen species, which includes nitric oxide, can be due to mitochondrial dysfunction, again due to damage to the neuron or other glial cells, hypoxia and glucose deprivation, right? We have neurodegenerative products such as beta amyloid from Alzheimer's, alpha-synuclein from Parkinson's, toxic metabolic products such as ammonium. So ammonium is positively charged. It is a byproduct of reaction with a protein, so just remove deaminases of glutamine to glutamate, as we mentioned previously. When the astrocyte cycles glutamate to glutamine, glutamine shuttles back to the neuron, and then a deaminase reaction will convert glutamine to glutamate and have a byproduct of ammonium. And this ammonium is extremely toxic to the whole body and will eventually be secreted by the kidneys. But if there's an issue, then this toxic metabolite will be present in the CNS, such. And also regulators of cell proliferation, such as endothelin-1. As we can see in these pictures, you have the normal functioning, uptake, release of synapses, nodes of Ranvier for the fibrous, and then the blood vessel, and also gap junctions between other astrocytes. And here are just really shows the insane amounts of molecules and the crosstalk between astrocytes and other cells and damaged cells, and just puts into perspective how extreme that this is. And this is not all of the signaling molecules, it's just a few, but kind of just goes to show the complexity of what we're trying to describe here to you guys on a high school and basic college level. Now, just more about the molecular triggers, but additions to these molecular triggers, and some more information is that, as I said in a previous slide, that these molecular mediators can be released by all cell types present in the CNS in response to any form of insult. So we talked about just now the complexity of this response in the crosstalk communication of astrocytes and other cells. This is important, right? This complexity is essential because this allows the astrocyte to react and be prepared for any type of CNS trauma or insult possible, and then have the best chance of responding the correct way. And the astrocyte, right, it's not able to look at a CT scan and say, oh, this guy has edema, or this guy has a stroke, which caused edema, or a GSW to the head. The astrocyte is not able to do that. All it's able to do is really say, okay, this is the molecular mediators I'm given, and that correlates to this type of response. So I'm going to give, or to this correlated cause. So I have this certain effect, which should be the proper response to that trauma. And finally, as we said just now, these molecular changes, these signaling molecules directly control reactive gliosis and how it changes the functioning and morphology of the cell. So what is the beneficial functions of reactive gliosis and glial scarring? Well, when there's more reactive gliosis, sometimes it can help and increase the uptake of this potentially excitotoxic glutamates, increase glutathione production, antioxidants, which will help sequester these ROS species and not prevent oxidation and not prevent apparent oxidation of random proteins and molecules, which can significantly disrupt the functioning of the CNS. Also, adenosine release for neuroprotection. Adenosine is really interesting because adenosine makes you sleepy. So it allows you to sleep more. It may put you in a coma, so your brain can have time to rebuild itself without having to worry about more trauma. And adenosine is the reason why caffeine makes you more awake, because caffeine will bind to these adenosine receptors but not activate it. So it'll block adenosine from binding and activating it. So if you have less signals to make you sleepy, then you're more awake, which is what caffeine does. So kind of interesting. And we talked about protection from ammonium toxicity because it is a toxic metabolite, so very important. Also, degradation of beta amyloid peptides in Alzheimer's, again, very important, so it doesn't cause accumulation in these plaques. Blood-brain barrier repair, which is very important, especially if there's a stroke, which can cause these endothelial tight junctions to be more leaky. This can help repair it. And as we said just now, right, reducing the vasogenic edema after trauma, stroke, or obsessive or obstructive hydrocephalus, which may be possibly seen in tumors and whatnot. Stabilizing the extracellular fluid and ion balance, which can help reduce seizure threshold because it reduces the amount of glutamate in the system, which can make sure that the neuron doesn't get overly excited and can fire off randomly, which can trigger seizures. Also, on the other hand, it can also uptake the potassium or release potassium and make sure that potassium levels extracellularly aren't too high or too low after a trauma. Also, limiting the spread of inflammatory cells or infectious agents to healthy parenchyma. This is more so related to the function of the glial scarring. So, the very dense collagen fibers at the extracellular matrix prevents the cytokines from going and affecting the healthy brain tissue when it should be more localized and should be functioning at the damaged part of the CNS. And finally, it can have pro or anti-inflammatory potential, which are context dependent. So as we said, the function of reactive gliosis is dependent on one time and also the distance. So typically in the literature, at the immediate onset of a CNS trauma, it will have pro-inflammatory mediators to attract a bunch of microglial cells and inflammatory cells to help try and contain it. But over time, chronic inflammation can cause cell death and is in the literature a proposed way that disease such as Parkinson's occurs. So after a while, it can switch to a anti-inflammatory response, which can prevent this chronic inflammation and thus neurodegenerative diseases. So now we are talking about the detrimental function of reactive gliosis and glial scarring. So one, this mainly refers to the glial scar formation, and this was the initial piece of evidence that mainly led all early researchers to discount reactive gliosis as a purely bad thing because it inhibits axon regeneration, which is very significant because if you have injury to the brain, injury to axons, after Wallerian degeneration, you aren't able to regenerate it. This is a very large issue. Right. There's also similar inhibition in the oligodendrocytes, if you guys remember from the embryology video. Secondly, as you said, it can have inflammatory effects and produce cytokines, which, if it's chronic, it can cause more damage to even the healthy tissue and can be a triggering factor for neurodegenerative diseases. It can also produce more increased amounts of ROS. This can be due to the effects of the inflammation. This can be due to the effects of mitochondrial dysfunction as a result of the reactive gliosis. So a big issue here. And as we said in the previous slide, it can uptake, but it can also release the glutamate, which can cause excitotoxicity and possible contributions to seizure genesis via the same pathway we talked about in the previous slide. Like I said, this goes to show that it can have beneficial function or bad functions depending on the context of the injury. So VEGF production can compromise blood-brain barrier repair because it can disrupt the endothelial CLN5, which will then promote its breakdown. So very interesting there. Additionally, it can have overactivity of these alkoporins, which can cause cytotoxic edema during trauma or stroke, as we talked about, because it can increase the amount of water coming from the blood vessels and can cause this edema. Cytotoxic edema can also occur because the hypotonic environment will cause cell lysis. Finally, it can also contribute to chronic pain. So yeah. Now here are my references list. So if you guys would like to learn more about it, if you have any questions, either email me or Sebastian, or you can, or we can look through these sources yourself and figure out for your own. However, I will let Sebastian now talk about CNS pathologies and the astrocytic contribution to those. So thank you.